I am Dr. Tamilselvi of Department of Microbiology at KSR College of Arts and Science for Women. Now I am going to present about the virology topic. First, the general properties of virus. Viruses are acellular in nature. That is, viruses are not made of cells and lack cellular organelles. They can reproduce only inside the living host cells and obligate intracellular parasites. Contain genetic material. Virus contains either DNA or RNA but never both. The protein coat. The genetic material is enclosed in a protective protein coat called a capsid. Nucleic acid is surrounded by a capsid. Some viruses also have an outer lipid envelope. They are very small in size, usually range from 20 to 300 nanometer in diameter, can be seen only with an electron microscope. In the case of host specificity, infect specific host tissues or cells, example plant virus, animal virus and bacterial virus, that is bacteriophages, lack independent metabolism, do not perform respiration, growth or protein synthesis on their own, depend entirely on the host cell's machinery. Virus can be converted into crystalline form outside the host, showing non-living characteristics. This is the crystallized nature. Shows both living and non-living characteristics. Living characteristics includes reproduction and mutate inside host cells. Non-living inert outside the host cells and can be crystallized. Causes diseases. Responsible for many diseases in plants, animals and humans such as COVID, polio and rabies. This is the structure of a typical virus. Its nucleic acid is either DNA or RNA. Capsid is protein coat. Then envelope is present in some viruses only. Viral replication. Viral replication is the process by which virus hijacks a host cell's machinery to produce copies of itself. The general cycle includes attachment, penetration, uncoating, genome replication, protein synthesis, assembly and release. In the case of simple flow diagram, attachment followed by entry, uncoating, biosynthesis, assembly, then finally the release. This is the bacteriophage lytic replication. First the bacteriophage attaches to the bacterial cell, then phage attaches and injects the viral DNA, host genome degrades, then viral genome direct synthesis of viral genetic material and the viral proteins, then bacteriophage's assembly, cell lysis and phage release is the final step. Steps in viral replication includes the attachment and absorption. Virus attaches to specific receptors on the surface of a host cell. Penetration. The virus or its genetic material enters the host cell. Uncoating, viral protein coat is removed, releasing viral nucleic acid, that is DNA or RNA. Biosynthesis, that is replication and protein synthesis. Viral genome replicates. Viral proteins are synthesized using the host cell's machinery. Assembly, maturation, that is maturation. Newly formed viral nucleic acids, proteins are assembled into complete virus particles called virions. Release. New virions leave the host cell by lysis, bursting of the host cell, budding, existing through the cell membrane. It's common in enveloped virus. This is a general diagram of virus life cycle. First is attachment, then entry, replication of gene expression, viral particles are there, then the assembly, finally the release. Then classification of virus. Virus can be classified in several ways. The most widely used system is the Baltimore classification, which is based on the type of genetic material and the method of mRNA synthesis. Other ways to classify viruses includes based on nucleic acid, DNA virus or RNA virus, based on capsid symmetry, that is helical, example rabies virus, isohedral, example adenovirus, complex, that is example bacteriophage, based on presence of enveloped virus, HIV, influenza virus, non-enveloped naked viruses or polio virus. Based on host, animal virus, plant virus, bacteriophage that infects the bacteria. Then common viral diseases includes common cold, influenza, COVID-19, measles, chickenpox, dengue fever, hepatitis B, AIDS. These are the common viral diseases. Thank you for